Hello everybody. Till now we have learned about the organization of CTD. Then in detail we have learned module 1, module 2, module 3, module 4 and module 5. Yesterday we have discussed about module 1 and module 2. Today we are going to learn module 3 in detail. So module 3 is mainly divided into drug substance and drug product. Today's session will be quality section of module 3, which is also called as CMC. So under CMC we are going to discuss about drug substance and drug product. So before going to drug substance, we will discuss about the DMF. So DMF is what? The drug master file, and it is a mandatory requirement that we have to purchase a DMF from a third party, and by using that DMF we are going to manufacture our drug product. And before manufacturing you have to see whether that DMF meets the US requirement or not, or if you are filing to Europe, then you have to see the CEP and the ASMF, that is active master file. Okay. Then what is the DMF review and why it is necessary, and what is the importance of DMF review, that we are going to discuss, and when does it become essential for a DMF review from the FDA perspective. Now why DMF is important and why it is called the DMF are not reviewed or approved. So we will discuss checking of CMC information and ensuring the appropriateness and suitability of the information defines the essence of DMF review. So it determines whether the information included in the DMF is adequate to establish that the material is suitable for the intended use. So DMF review becomes an important process of an approval of IND, NDA or ANDA, and it is required to ensure that DMF is adequate to establish the use of intended use in the finished product formulation. So coming back to same question, why DMF is never approved or disapproved? Because when an applicant who is going to file generic application ANDA, that applicant has to purchase a DMF from the third party, and he has to give one LOA, that is called letter of authorization, to health authority. So based on that letter of authorization, when FDA will review your application, that is ANDA, that time the FDA may revert back with his own questions about the DMF. If he has any queries regarding the DMF, whether the impurity levels are high or any kind of queries which comes to the FDA's mind, he can directly ask to the DMF holder. So while compilation of ANDA or the generic application, you have to give the letter of authorization to FDA, and based on that letter of authorization, FDA may revert back to DMF holder as well as the applicant. Why this is important? Because DMF is never approved or disapproved alone. It will be open only when the ANDA application will be reviewed, and that is why it is called DMF is never approved or disapproved. So as I said, CMC is divided into drug substance and drug product. So under this drug substance, again it is divided from S1, S2, S3, S4, S5, S6, S7. So before I discuss all these parts into detail, what we have to see, I will give one example of roti, how to make a roti. So how to make roti? When we want to make a roti, you have to take wheat flour, which is your active pharmaceutical ingredient, in which you are going to add water plus salt plus vegetable oil So each ingredient has its own function. Water may act as a binder, salt may act as for the taste, and sometimes it may act as a preservative. Vegetable oil may act as a lubricant. So when we select wheat flour, we have to select the good quality of wheat flour, correct. Nowadays we get the ready made atta, that is Ashirwad atta or Pillsbury atta, but in old days we used to select the wheat, actual wheat, whether it is a surti wheat or lokwan wheat, like that. So there are different types of wheat and wheat flour available in the market. So when you select the wheat flour, which is considered as your main active ingredient, which is an API, to that you are adding different excipient. So each excipient has their own function. Water acts as a binder, salt acts as a taste or the preservative, and vegetable acts as a lubricant. When we mix these all ingredient along with API, that is called manufacturing process, correct. You mix it, then you form a dough. When you form a dough, then the next process is you make the small balls out of it, then you make the balls and then roll it. Then once you make the chapati, then you have to roast it. If you are roasting, you are controlling the flame. If you make the flame high, roti will burn, right? It will be burnt. Or when you make the roti, the flame low, it won't be fried, right. So the final output is roti, correct. But during this you are selecting API, excipient, then manufacturing process, then here you are optimizing the process. When you are roasting, you are optimizing the process, optimization of process, and the in process control, correct. You are controlling the flame, correct, whether it is high, whether the roti should be roasted on high flame or the low flame, that you are optimizing. So that is called in process control. Once you make the roti, that roti can be packed into different packaging materials. Like in our case, we generally pack into Milton or Tupperware, right, or you can pack into alu foil as well. So there are different packaging material wherein you can pack your roti, correct. Then once you pack, what happens? It will remain stable. So you have to conduct the stability. So you have to conduct the stability data. So similar things, if you apply the logic in your API and drug substance and drug product, how do we collect the data for drug substance plus drug product? So drug substance part is divided into S1, S2, S3, S4 and S5, S6 and S7. So each section now we will discuss by considering example of this roti, but the basic concept remains the same. The first step is API characterization, then what? Manufacturing, then optimization of sweet, then in process control, then the packaging material, stability, correct. In between I have missed one step, that is reference standard. So when you make the roti, that roti has to be tested, right. When it is tested, you always compare with the reference standard. So when you are at home, your husband will say, आपकी रोटी मेरे मम्मी जैसी नहीं बनी, correct. So that time he always refer to one reference standard. So that is why reference standard and the test. So your roti will be a test and it will always be compared with the reference. So you are clear with the concept how it is applicable in our drug substance and drug product. Once you make the roti, roti will become your final product. Then for this roti also, you have to gather information in the form of P1, P2, P3, P4, P5, P6, P7 and P8. Now the main thing you should keep in mind, that is drug substance part, that is from S1 to S7 is coming from the DMF holder, because you are getting DMF ready, correct. So in that DMF you should see to that all the sections are ready, all the sections are nicely written, and those sections has to be justified. Okay. So being a regulatory professional, you have to review the DMF parts thoroughly and see to that whether that DMF is helpful as per the rules and regulations, and whether it meets the requirements of US FDA or Europe or wherever you are going to register your product. Now coming to P part, correct. P part, in P part we have to discuss from P1 to P8. So same logic if you apply. What is P1? P1 is a composition, P2 is a PDR, P3 is manufacture, P4 is what? P5, P6, P7, P8. Manufacturing process, then excipient, then control of drug product, then reference standard, and then container closure system, that is packaging material, right, container closure system, then the stability. So in drug product also, if you see, more or less the same sections are there except the P2, that is product development report. If you compare with S1, S2, S3 up to S7 and P1 to P8, more or less all the sections are same as per the drug substance. So similarly you have to gather all the information about your drug product. So drug product is manufactured in our laboratory by the pharmacist, and it is analyzed by the scientist, the analytical scientist. So all the data has to be collected from different stakeholders, and you have to write PDR, that is pharmaceutical development report, as per ICH Q8. ICH Q8. Once PDR is written, then it has to be reviewed by regulatory professional. Okay. So similarly all these documents are coming from different departments, are reviewed by the regulatory professional, and then it is compiled in the format of CTD. In that CTD, under CMC, drug substance and drug product. Clear? The concept is clear? You must have realized how the logic should be applied from the example of roti. Okay. In roti also we are following the same steps right from the drug substance to drug product. So now we will discuss each and every section from S1 to S7. So in S1 is again divided into S1.1, that is nomenclature, S1.2 that is structure, and 1.3 is general properties. This section is very important, though it is called general information about drug substance. Why it is important? Because during development the information about the polymorphism and stereochemistry, including supporting information apart from other general properties, must be a part of general properties. A description on solubility based on different pH buffers, that is pH 1.2, 4.6 and 6.8 has to be provided. pK value will have to be included. So these are the special checks we have to consider while gathering the information about what? Drug substance under general information. Though it is called general information, the molecular weight, the structure, the API characterization data and the polymorphism, solubility, pH value, pK value, everything is important, because it is going to affect your quality of the product at the end of the shelf life. And that is where you have to see, when you review the DMF, you have to see whether the vendor has captured all the information under the general information or not. And then let us talk about S2 part, that is manufacture. Manufacture again divided into S 2.1 to 2.6, which again gives the manufacturer information, that is who is the manufacturer, his name, address and everything, then the description of manufacturing process and the process control. Suppose if your manufacturing process has 10 reactors, 10 steps, then it has to be described in detail, that is step 1, step 2, step 3 up to step 10, and in between if he must have used any reagent, any activator or anything, solvent, and everything should be mentioned under the manufacturing process, and the manufacturing flow chart has to be given. Then what are the controls, has to be means what are the controls considered during that manufacturing of the drug substance, that you have to give into the write up. Then the critical steps and any intermediates. Critical steps are again very important, because sometimes what happens, we forget to write the critical steps and that becomes the deficiency. So critical steps are very important. Again the DMF is divided into open part and the closed part. So critical steps are already, you should mention into the closed part, that will not be a part of open part of DMF. So that you should keep in mind. Then the process validation and evaluation, whatever manufacturing process you must have developed, how it was developed, how it was validated, and the development summary report along with the process validation report you have to provide into this section. Then so this is all about the S2, but during this write up of S2 part, what special checks have to be kept in mind, that is, first is route of synthesis should include the intermediate name, reagent, solvent, catalyst used in the synthesis, then flow chart to capture the in process checks. In case of advanced intermediate, the chemistry of the same must be included. Then the manufacturing process write up should be simple and should be similar to the actual process. Over detailing may be avoided. So generic write up should be followed in the open part of the DMF. Reprocessing criteria should be defined. Usually reprocessing is avoided, but if at all you are using, then it should be justified. Then batch size details and the batch formula to be clearly defined with the scope of the minimum and maximum batch size captured, only in case of restricted part of the DMF. The control of materials should be complemented by the supplier and the in-house certificate of analysis. So critical steps should be checked with the process development report and must be correlated. Further, a proper justification should be in place for classifying them as critical. This information must be present in the development report. If at all you are saying that this step 5 is the critical step, you should be able to justify why it is critical, right? Otherwise it won't be acceptable by any health authority. So you are the development guy, you are the manufacturing your drug substance, then should be aware of each and every step of the manufacturing process and how it was developed. Then the intermediate specification must be justified in terms of purity, related substances, residual solvents, catalyst. A mass balance must be existing while defining the specification of the intermediates. For key starting material, the above approach should be followed. What is key starting material? The key starting material plays an important role when you start your manufacturing process for any drug substance. So key starting material, if you are unable to define properly and if you supply all the relevant documents received from the vendor who is supplying your key starting material, then you will face a problem in future. Then the process validation protocol and report has to be correlated with the batch manufacturing records and must be verified for all in process and critical parameters. Critical parameters should be captured in the process validation protocol and must be connected to the development report. Each product you must be aware of that process validation plays an important role and you have to take minimum 3 batches as a process validation. because of the consistent result, right. Once you get the consistent result, then you can claim that your process is validated. And how it was validated? It has to be documented by the mean of PVP. The process validation protocol has to be in place, and then only you can go for process validation report. So, process validation protocol and report should match, and if there is any difference in the process during the validation process apart from your protocol, then it has to be justified. Generally it should not happen, there should not be any difference. Then the manufacturing development report should contain the rationale of the synthetic route, should cover the genesis of impurities, the use of appropriate solvents, and the scientific justification of defining critical steps and defining intermediates, proper experiments, negative observation to be conducted for process optimization. Formation of the right morph and the right isomer, which is usually covered, must be a part of your development report. This precaution has to be taken right from the beginning, whether your product your substance has the polymorphism, isomerism, or the solubility issues, pH issues, that has to be considered during the development stage only, and proper justification to be given, and everything should be captured in the manufacturing development report. Then S3 talks about the characterization. Here you have to capture the structural elucidation and the impurities. What are the special checks? That is, proper scientific information to be provided for the polymorphism and identification of the stereochemistry of the active ingredient amongst other special studies, that is UV, IR, NMR, mass balance, DSC and XRD. DSC is differential scanning colorimetry. So, these are the special checks under the S2, S3 characterization part, the section. So, impurities, again it is very important, and impurities has to be analyzed as per the ICH Q3. And apart from the normal process impurities, residual solvents and degradation impurities, impurities due to starting material, when the key starting material is in an advanced intermediate, should be included in the write-up. So, impurities are mainly generated due to the key starting material, then the reagents or the solvents used during the manufacturing process, then during the stability also impurity may be generated, that is also called as a degradation product. So, whenever at which stage the impurities are being generated, that you have to capture in the write-up. Then absence of a catalyst if used should be provided. Any special reagent like complex compound should be captured in the impurity write-up. So, this impurity section is very important, and how effectively you write that section is based on your skill set again, because after writing you should not get any queries from the health authority, that precautions to be taken by a CMC writer. Then a mention on genotoxic impurities has to be captured in the impurity write-up. A proper write-up indicating that route of synthesis does not involve any formation of a genotoxic substance with proper justification, that is reference of structural alerts and Ames test. So, Ames test is generally carried for the genotoxic impurities. Nowadays, as per ICH M7, these genotoxic impurities are also becoming a most important part of any dossier. Why it has become important? Because of any answer? Because of the safety, because any health authority's main objective is to protect public health, and you have to meet those requirements, and you have to prove that your product is of safe use when it is consumed by a human being. So, the impurities must be also appropriately captured in the specification of the final product. So, till now we have discussed S1, S2 and S3 part. Now we will discuss S4, that is control of drug substance. Under this section you have to attach a specification, method of analysis, analytical method validation as per ICH Q2, batch analysis and justification of specification. We will discuss each one of them and what are the special checks to be considered under this section. So, specification should include test parameters which sufficiently controls its quality, and a good enough specification is actually not good enough. Example, minimum 2 identification tests with one as stability indicating and specific. So, you have to include 2 identification tests, one is by HPLC or any instrumental analysis, and the second one could be a chemical analysis, IR spectra or UV spectra. So, that is a mandatory requirement when we go for the regulated market and under the ICH CTD sections. So, all known impurities, single unknown and total impurities needs to be controlled. An XRD test is a must should the molecule exhibit a polymorph. Catalyst, if any, used in the synthesis of API. Key starting material may be controlled. So, not necessary if absence in 3 batches shown, and residual solvents need to be included. Assay and related substances will have to have a stability indicating method, although the compendia method may be a titration. Why I have written like this? Because your assay method or your product could be an official in USP, but USP says that the material is analyzed by titration, correct. Still you have to develop your in-house method which is HPLC and which should be stability indicating. Why it is called stability indicating? Because during the stability many degradation products are generated and your method should be rugged enough to capture those impurities during the analysis. So, you have to develop a method and validate as per the ICH Q2, which is called the stability indicating method, because that method will take care of the impurities or the degradation product generated during the shelf life. A proper stability indicating chromatographic method will need to be developed for a TLC test, although the compendia may recommend a TLC test. If it is a TLC method recommended by USP, then also you have to validate that method. The method reference, that is compendia or in-house, should be included in the specification page of DMF. The method validation for key testing parameters like assay, related substance, residual solvents, metal catalyst should be adequate and conducted as per ICH. Specificity, precision, linearity, range, robustness, accuracy, force degradation, solution stability, etc. will have to be a part of validation package. So, for the validation package you should refer to ICH Q2 guidances, the most updated guidance. The method validated should be the same as that of the final method adapted to test the drug substance, because you are taking the DMF from the third party or the vendor. So, your method should be similar to the vendor, it should be adapted from the vendor, from the DMF, and it has to be validated at your site in your factory. Then the similarly when you validate the method for drug product also, you have to adapt the method for the drug substance, you have to develop along with your drug product and the placebo, and then you have to validate that method as well. The certificate of analysis should have the batch size mentioned among other typical details. Typical chromatograms may be provided for a particular batch of API. A scientific rationale has to be provided for the specification adapted for the drug substance. This is more critical if the molecule is in-house molecule. Thus, all testing parameters have to be justified properly and should have scientific bearing. Let us talk about S5, that is reference standard. In this, the special checks are in case an internal standard, that is primary reference standard, is used, then the method of preparation of the same should be included with the proper structure elucidation details. The qualification details should be clearly stated in case of the working standard is qualified. Many times what happens, reference standard are very costly, and because of the cost we are unable to use it for the routine analysis. So, based on the reference standard received either USP, EP or BP, you have to make one working standard for the routine analysis and you have to qualify. How do you qualify? You have to
by testing reference.
Yes, test with the reference and generate the data, and whatever data is being generated, the analytical data, the chromatogram has to be attached under this section, and you have to give the comparison between the reference standard and the working standard. So, the COA of all working standard needs to be provided with typical chromatograms. In case of reference standard use, then the proper lot number needs to be given. Then S6 is a container closure system. Special checks, that is IR spectra of the polybags, needs to be submitted. Now, in case of drug substance, you are going to pack into the polybags, whereas in drug product you are going to pack into blisters, HDP jar or alu blister, whatever the bottles and all. But in case of drug substance, the polybags are used and then those polybags are kept into the big drums. So, when the material is coming in contact with that polybag, so how the quality will be affected or how it will be stable, that we have to prove that after storing into the polybag, still our drug substance is stable. So, you have to provide the specification about that polybag, that is IR spectra, specification, COA, the material safety data sheet. So, all these documents are important when you are purchasing a polybag from a third party or the vendor. So, the supply detail consisting of the compliance certificate, then the declaration as per current European guidelines, compatibility of the polymer granules from the manufacturer needs to be given. Generally these polybags are made up of polymers, different polymers are used. So, this is very important, and the material of construction is very important. The supply declaration is also has to be given. Then safe use of plastic material and articles intended to come in contact with the foodstuff or pharmaceuticals or drinking water should be provided in this section. Then the last section is S7, that is stability data. So, this section is also divided into 7.1, 7.2 and 7.3. So, 7.1 is the stability summary and the conclusion. 7.2 is post approval stability commitment you have to give under this section, and you have to attach the stability report which you are conducting for the accelerated and the real time shelf life. So, what are the special checks in this section is to check the stability specification against the test protocol, that is stability protocol, then the stability condition and the stability time points, check parameters like polymorphism etc., and the post approval stability protocol and commitment should be in place. Why this commitment is important? Because during the application you are providing only 6-month accelerated stability data along with the application, and you have to give the commitment that further stability studies will be conducted and the report will be provided as an annual update or the amendments. So, stability data is generally 6 months for the US submission, 6 months for the US submission and the 6 months for the Europe submission as well. This is I am talking about 6 month accelerated, that is 40 degree centigrade and 75% relative humidity. So, storage condition and retest date should be included in the stability summary, which should be supported by the stability data. Earlier we used to submit 3 months accelerated stability data. So, that is why I have corrected this slide. Then module 2 we have already discussed, and module 3 is also covered. Under module 3 we have covered only drug substance part, that is S1, S2, S3, S4, S5 and S7. Thanks for your kind attention.